Well, good afternoon, everybody. My name is Colin Hill. I am the CEO and co-founder of AI virtual patient company, GNS, and also a board member of pain therapeutic company called Centrexion. And I'm really pleased to be moderating the session today with my esteemed group of colleagues and partners, representing both really the top of academia and the top of industry. Now, I'm going to start off with some opening remarks, and I'm going to have each panelist introduce themselves and give their opening salvo. So first off, this is a rather provocative title, why the FDA will bet on in-Silico simulations to really drive the creation of new drugs. So let's unpack that. What does that mean? We've been living through really explosive growth in data and compute power in AI that's now enabling a different way of running clinical trials. Right? And starting to point to the opportunity to potentially model and simulate the clinical trial before running the clinical trial. Now, this concept in computational simulation for product development is not new in most industries, right? In fact, it's been around for decades in the aerospace industry, the auto industry, certainly computer chips and the like, but yet here in the discovery and development of new drugs, we have not yet been able to bring these methods to bear. And it's not disconnected from the fact that we have, compared to these other industries, probably the highest product development failure rate there is. And at the core of it, of course, is the complexity of human biology, the complexity of human disease, that after decades and decades of molecular biology research, we're lucky if we know 5% of the circuitry of human disease on a genetic level. That means we're missing 95% of that circuitry. And now that we're living through the omic wave, coupled with massive advances in computing and AI, it's now possible to think very differently about how we can approach clinical trials, how we can simulate trials ahead of time, as opposed to a trial and error approach. And what we're aiming for is the concept of finding that magical population of responders versus non-responders in the trial. Can we get there? And I want to make it clear, we're not talking about just clinical trial operations and site selection. It's a different problem. This session is about really going for the brass ring of being able to truly ingest massive quantities of data, including molecular data, to produce virtual patients and actually run simulations of trials. So with that, as an opening framing, I'm going to call on my colleagues to go a bit further on really their opening positions. And we're going to start with Steffan Ho from Pfizer.
Well, thank you, Colin, and thanks very much for the opportunity to be on this panel. It's a great topic for sure. My name is Steffan Ho. I head up what we call Translational Oncology at Pfizer in our global product development organization. And what does that mean? There's translational everything. What that means is we are focused on ensuring that as we attempt to move drugs forward into the clinic to generate evidence to bring them forward for all of us, for patients, we prioritize our efforts to ensure that our strategies are driven by the science. So that's the bottom line. It's the science that dictates how we develop our clinical trials, what patients we move them into, all the way going back into the preclinical setting, what targets we think are going to be most productive in terms of interdicting in the relevant diseases. So that's really the focus that I have. By way of background, I'm a pathologist by training, so historically I've trained looking at the disease process under a light microscope, so I'm incredibly excited by the opportunity over my career to go beyond the light microscope into the molecular characterization, and now actually with digital spatial profiling, you can go into an extra high power objective and even get molecular characteristics in a spatial context. So for me, I think it's just incredibly exciting. You know, with the platforms we're applying now to disease, we have this comprehensive view of the biology, but the critical challenge is with all this incredible data and all this comprehensive view of the biology, how the heck can we actually utilize that to make an impact on this objective of bringing new medicines to patients? So I'll stop there and we'll continue the discussion.
Fantastic. Sid, please continue.
Okay. Thanks, Colin. Thank you, everybody. It's a great opportunity to be here and take part in this very exciting panel. like Steffan and Colin mentioned, the topic itself is something I find very interesting, and not only that, it has major implications for us even in a shorter term than we think it might happen. So I'm a molecular biologist by training, went into drug discovery and then development through bacterial infectious disease, immunology, and then now immuno-oncology. So my aha moment came when I was leading multiple programs at my previous place of employment, which is Amgen, where we were working on immunotherapies and saying, well, how do we take this to the next step? And my initial thought about artificial intelligence, and everybody's talking about that, is that I don't have enough real intelligence to understand artificial intelligence, so just leave it aside. But the realization, the aha moment came when we were dealing with a bifunctional molecule, which has a developed based or designed based on an anti-PD-1 monoclonal antibody backbone attached to a cytokine in a very cleverly designed molecule. So we have a cis-acting activation of the cytokine involved. And the key question was, where do you take this drug? To which patients, which line of therapy, what combination or not? So I was thinking, well, first of all, PD-1s especially have changed the treatment paradigm in immuno-oncology, as you all are familiar with, in multiple tumor types. But the reality still is that many of our patients don't really either respond or don't respond in a durable way. And obviously, there are reasons for that. Our understanding of predictive markers to say, well, this patient is more likely to respond versus that is at a very basic level, I would say. There's a whole lot of room for improvement. Number two, to then say, well, for this patient, I should add on top of PD-1 a particular cytokine or another checkpoint. Understanding what predictive markers would allow us to select those patients or enrich for those patients, that's also, the understanding is at a very basic level or non-existent. And then with all the development in multiomics and assays and methods and technologies, it became clear to me that manually we cannot deal with all these data coming from different points. And if we can't analyze them in a proper way, leading to probably composite biomarker signature rather than a mutation on wild type kind of a binary thing, we won't be able to take the right therapies to the right patients. And why it's important for the FDA or any health authority, that's the path to making sure our trials are more designed for the patients who are more likely to respond. That will lead to better probability of success. That would lead to better approval with more robust data, and will shrink time. It's a win-win for everybody.
Fantastic. Andrea.
So I'm Andrea Souza with Eli Lilly and Company, amazing organization to work for. And in the context of what you just heard from a translational science-driven approach to actually a very specific predictive biomarker approach where we tend to fail in virtual digital patients, I thought I'd quickly share a story. Steve Ruberg is a statistician that worked at Lilly with somebody else who currently still works at Lilly, Lei Shin. And they wrote one of the first original papers around this concept of virtual patients back in 2011. So we do believe in the usage of synthetic data, but to the point Colin's making, it's still early days, and there's a lot more to figure out in the age of AI and getting to that stage where we're using it across clinical trials for much more than trial selection, as you put it.
John, representing academia.
Well, I'm glad somebody gets to carry that burden today. But I want to first say it's really an honor to be here with old and new friends on this panel and in the audience today. And I'm actually very excited to talk about this topic. And I want to riff a little bit on something that Colin said earlier, actually two things. I'm a professor and chair in the Department of Biostatistics at the Harvard Chan School of Public Health. I'm not a statistician. My PhD was actually in theoretical physics. I do computational and systems biology, so I do lots of things I'm not supposed to do, which makes it fun. But what I really like about living today in the world that we as scientists and people interested in drug development live in is that we're actually in a really data-rich environment. And I think of data as the raw material that we all want to consume to be able to build models that we can use to better understand the world. And as Colin suggested in his opening, really sort of predict what's going to happen based on building models that take advantage of the computation that we now have available that we couldn't even dream of a few years ago. Right? So we have this really interesting opportunity, but biological systems are fundamentally complex. And the example I like to point to most often is if you look at the 250 or so different cell types in your body, each one of those cell types has the same genome, the same set of genes, the same set of mutations, but each one carries out a different program and responds differently to external perturbations, different threats. So your brain cells are making neurotransmitters, your liver cells are making digestive enzymes, and there's a reason. And the reason is that they're complex networks that are active in those cells that are influenced by genetic, epigenetic, and environmental factors. A lot of the work that I and my colleagues do consume large data sets and try to model those different interactions. And we published a paper a few months ago that I think represents the first demonstration of the fact that we can make predictions about differences in regulatory networks with the same individuals in different tissues that are disease relevant. The challenge we have, though, is to take all these complex models and synthesize them in a way that it gives us understanding of different responses that my colleagues here would like to try to model in testing drugs. And I think that's a challenge, but also an opportunity, because we really have the opportunity to pull and synthesize data by integrating things rather than taking a reductionist approach and really looking at individuals and organisms as a whole. But we also have a great opportunity to try to link information together in ways that will drive trials forward.
So John, an edgy question. Do we need to understand the full complexity of the biology in this data-driven way in order to accomplish the very practical needs of better patient selection in trials? How far do we have to go down the systems biology, mechanistic understanding for Steffan and Sid and Andrea to be able to design trials that get the benefit of that enrichment?
So I think that's a great question. And I think it's one probably everybody here on this stage wrestles with at different points in time, right? You can describe a ball falling off the top of the building here and hitting the ground without having to invoke Einstein's theory of general relativity, even though we know it's all gravity, right? We have biological systems where we can make observations and build phenomenological models that will really help us understand and make better predictions without understanding all of the complexity. But it kind of depends on the question we want to ask, right? If you think about drugs that we're trying, it may be some metabolite that's generated in the liver that affects outcomes or causes side effects that we didn't expect. And so I think we have to be ready to sort of shift back and forth in our perspective depending on what we need to do to get the best answer for the questions we're trying to address at a particular moment in time.
Yeah. So one key question is, where are we on this journey? I think we all agree we are on a path towards this becoming a reality. We've had major advancements in data. It's happening more and more every day in terms of rich clinical genomic data sets across diseases, whether it's oncology or Alzheimer's or other types of areas. Computing power has become so fast and so cheap, we don't even talk about it much anymore, right? We take for granted the fact that we can plug into cloud computing resources. And of course, you can't pick up a book or look at anything about this industry without hearing about AI. And so the question is, where are we on this journey? Are we at the baby steps level? Are we already really in the middle of it? Or is this about to become something that both industry and the FDA start to embrace in a big way? And what are the limiting factors? And so, Steffan, I'd like to start with you there with maybe a special focus also on the data piece, because I know a lot of your focus is generating that rich, high throughput data.
Yeah, no, all good questions. Where are we on this journey? I think part of the challenge for me is we throw out these terms AI and ML, and they can mean a wide spectrum of different things. Mm-hmm. So that's one key thing, is what exactly do you mean by AI, machine learning? Is it deep learning, neural networks? That's a whole different ball game compared to other applications of so-called machine learning or statistical learning that actually have proven themselves. So part of it is we need to be very specific about where we are. So to answer that question, I would need to understand, okay, what exactly do you mean by AI and machine learning or in-Silico this or that? So that's one perspective. But getting to the key point is, again, depending on what you're trying to do, and I think it all depends on what is exactly is your objective. From my perspective, it's ultimately to develop a clinical test that can be used to identify patients for treatment selection. Patient stratification, that's the premise of precision oncology. A diagnostic test that you apply to a patient, and that then determines how you will treat that patient. That's what precision oncology is. In my view, we've been very successful in the first generation of precision oncology. Single analyte diagnostic test, dominant driver oncogene biology. So proven, continues to have incredible advantages for patients out there, and will continue to have that advantage. But the bulk of disease, in oncology at least, is much more complex. And there, I think we still haven't proven, we don't have proof of concept that we will be successful in applying this premise of precision oncology at this next level of biologic complexity, as you're referring to. So for me, I would say we're not there. It's an open hypothesis in terms of multimodal omics data. Can we bring it all together to actually develop a truly clinically applicable diagnostic test? And I emphasize that because it's one thing to generate a model. It's another thing to reduce it to practice in the clinic in terms of robustness and technical validity and accuracy. So for me, it's an area where we are not there by any stretch of the imagination, but it's an area where we need to focus efforts and intensively understand what are the models, what are the approaches, what are the limitations of the data, how should we design these efforts to actually get to that end result, and keeping that end result in mind as we move forward.
What's the biggest bottleneck in your mind?
The biggest bottleneck, and I get this from some of my data experts, is well, they call it the curse of dimensionality. And my understanding, the comparison is this, and here's a nice one. So we've all heard about Netflix, right? Yeah. They do all sorts of amazing things with AI because they've got 18,000 movies and 500,000 people, and they can they can determine what all these 500,000 people like in terms of these 18,000 movies, and they can predict it. They can predict what you want to watch, and they'll tee it up for you. And so that's great. And the whole point is, okay, let's apply this to what we're doing in the life sciences and cancer biology. But the problem there is the biggest database, for example, the Cancer Genome Atlas, the US government-funded catalog of biologic complexity of human cancer, maybe 100,000 patients in that database. And what are the number of variables? Say 3 billion. So you can see it's just in multiple orders of magnitude much more complex, even compared to the successes we've had. So that's the problem. So we try to categorize and develop supervised clustering of data. Inevitably, most of them are wrong, and they don't validate because there's so much false discovery. So to me, that's the biggest challenge. How can we sift through this data, apply the kind of applications that John and Colin, you guys are developing to address this broader problem, reducing the dimensionality and so on and so forth. I'll let others speak to that in a more informed manner. But for me, we've got a long ways to go. There's a lot of infrastructure challenges in terms of the data. I work with the largest clinical trials in the world, in cancer at least. 600, 700, 800 patients in global clinical trials, randomized controlled data. that is small data compared to what one would need for some form of a
Yep.
deep learning application. So part of it is to realize what the limitations are, and then be very clear-eyed. Focus on the limitations, and don't just keep applying new algorithms and say, "Oh, isn't this interesting?" Keep an eye on what we're trying to accomplish, because I think one challenge is that with the hype of AI, if we keep saying, "Oh, AI is the answer, ML is the answer," and throw these terms out there, and then we apply them to no success, that ultimately is going to be detrimental to the field. We need to have a clear idea of the limitations, and then every step we take should be focused on how are we going to overcome this well-understood limitation. And I think a lot of it now is in the research phase. We've got people that are incredibly bright at understanding the data and the algorithms to really try, apply these approaches. And most of them won't work, right? But I think we can't just apply them because it's the next new thing. We need to apply them with an understanding, here is the challenge that where all the prior approaches have failed, and actually recognize that some approaches are not applicable yet, and then turn our creative energies into overcoming the critical challenges as opposed to just throwing AI, ML, the next new algorithm onto the problem and seeing what happens.
So very good. So I sum up what you just said as we need bigger data. And we need to be very clear and focused on what we're solving for in getting to better patient enrichment.
Yep. So I'll confirm and then gently agree with some parts and disagree with others. So, and use an example. I do think it's early days. I do think there's tons of work left to be done. I do think you need data, but more than data, you need exactly what you said. What business problem are you trying to solve? What's the scientific question of interest? Here we're talking about find the patients, find the right patients, and find the right patients faster, right? Because clinical trials is really, really expensive. But I do think there are advances being made with AI and small data. So our scientists in oncology recently gave us a very tough problem, and it's for the RET alteration. Not thyroid, where you've got the point mutation, but actually lung cancer. And so we partnered with a very hungry, young, innovative company, Pathology Images, and we're able to meet the criteria of the proof of concept, right? And what was the criteria for the proof of concept? We typically have to screen 50 patients to find one patient currently. And the way sequencing is done, you look for all of the frequently occurring mutations before you eventually get to RET, which is less than 2%. So often you might actually be out of time, and you might be out of sample. So there is a potential big win if right at the pathology exam, you could have a detector that says it looks like this is the mutation, right? Understanding complex biology. So our criteria was find all the positives, because we didn't want to lose any of the patients, and make sure at least 50% specificity is what we had. And we met the criteria. The name of the company is BioAI, but they are true experts in what they do, and we worked closely with them. Lots and lots of more work to be done before we get that type of an experiment into clinical practice. But there are advances with data, with compute, that make this the right time for us, industry, academia, partnerships to come together to bring this to fruition.
Just a clip, that's a digital imaging application, so you're looking at H&E images to try to enrich for RET positive?
Yep, H&E images, and basically they QC'd really well. It was an ensemble model. They did have human in the loop, so they had pathologists actually annotate, and they haven't completely shared the details of the model, but we held back a set of images and then gave it to them, and we knew which of those were positive or negative because we wanted to make sure that we were testing them. And it was a bake-off. We baked off 3 different companies before we realized they had figured it out. Extremely, extremely difficult problem to solve.
If I could add a little bit to where Steffan was going with what's the gap, I would also add that our understanding of diseases or our definition of a disease is still not correct from a pathophysiological perspective. So calling somebody having non-small cell lung cancer or basic histology-based difference, it's good, it's not nothing, but it's not where it needs to be. So the more we understand that we are calling something a disease where my true mechanism might be suitable for only a segment of that, and that also may not be defined, again, in a binary way through mutations. It works in driver mutations, but not everywhere. So this enrichment that my likelihood of responding is higher if I had markers X, Y, and Z, and it may not be one marker. That would be one. I would also say is that the more we recognize our definition of disease is faulty, we have to get away from, oh, here's a new mechanism. Somebody said, let's try non-small cell lung. Somebody, oh, pancreatic, it's a very high unmet need disease. So why should we study it? Well, it's a high unmet need disease. Well, how does this mechanism fit there? Oh, well, we'll figure something out. So the way I think true, very conventional part of clinical drug development is working needs to be improved, and there's some translation of language spoken by more the clinicians who are running those trials and the biomarkers. That is also a need from my perspective.
John?
So, the Netflix example is a good one, right? If you think about it, because there's a low stakes game, right? If Netflix recommends a bad movie, you waste 5 minutes and it's not a big deal, right? If we have a multi-million-dollar trial fail because we've had bad patient selection, it's a huge deal, right? And it also puts patients at risk. So we have to think smartly about how we solve this problem. And again, Netflix is a great example. If you take all movie titles and just throw it in and try to get people to recommend things or get an AI system to recommend them, it's much harder than if you reduce the complexity of the problem. How do you do that? Well, the way you do that is you recognize that some movies are comedies and some are dramas and some are action, right? And you start to put them into groups.
Break it down.
And then you start to think about who the actors are and who the directors are and who the writers are. And so pretty soon you go from thousands, hundreds of thousands or tens of thousands of movies down to a smaller set of parameters that you can actually use to try to make recommendations, right? And the challenge is reducing the complexity of the problem in an intelligent way. My favorite paper in all of computer science is the 1997 paper by Wolpert and McCready called No Free Lunch Theorems for Optimization. And they talk about this very problem, that if you have a really complex problem that you want to solve, the best way to solve that problem in finite time and get arbitrarily close to a near optimal solution is to start with a guess, right? You take the data and you reduce the complexity by bringing in the biology making intelligent guesses. And the guesses we make in Rett syndrome may be different than the guesses we make in immuno-oncology for how to reduce the complexity. And that's where expert involvement in trying to develop these systems really becomes important, right? It's because we have to think about this. Sort of going back to the question Colin asked me earlier, though, do we have to consider everything? Well, if we're thinking about doing this, right, it's like, I love the title, or in the title we have the word bet. If you were to go to Las Vegas and you could only get a few % advantage on the house, you're going to go and gamble because in the long term, you're going to win, right? And what we have to do is iteratively optimize the methods that we use to improve the odds of success, because in the long term, that's how we're going to win.
So we're going to switch gears for a second and talk a bit more about the regulators and where they are and where we think they're going. But we're also going to open up the floor to questions, not just at the very end, but during. So if there's any questions, please put up your hand. Otherwise, just walk up to that microphone so you get captured on the video. So external control arms have become becoming more and more standard in clinical trials and across a number of therapeutic areas. So the FDA is clearly showing an openness to considering data from these sort of synthetic patients. Where do we think the FDA is going to be in the next 3 to 5 years on not just considering data for the control arm, but even getting to biosimulation of the efficacy arm? To what extent do we expect the FDA to start considering data that's not meant to replace the trial, the efficacy arm of the trial, not in the next few years, but to be used to better optimize the patient selection? So where do we think the FDA is and where are they going? I'll start with Sid.
Sure. So my thinking on this is it's a situation, if we don't build, nobody will come. So the onus is more on us to start that conversation, and that requires this collaboration. It cannot be done just by industry, just by academia, just by government. We do need that involvement, that this is a collective win for all of us. Let's start with safety first. Let's also recognize that when it comes to pharmacokinetics, people don't think, why are you doing simulation? That's like a standard practice now.
Yeah.
Shouldn't this be applied to efficacy prediction as well? So I would go into safety first, run these simulation trials, see how successful they are in predicting the real conventional trial behavior. Get to the maximum tolerated dose, efficacious dose, and also look at pharmacodynamics and model those that are more likely to be predictive of response and efficacy. Take it to phase 2, and again have a synthetic arm combined with real arm. And then I think we can have a real substantive conversation about the registration, which is where the real win is. That would be my way of doing it.
So Sid, why safety first versus efficacy? Is it easier? Is the FDA just more open to it?
I would say as a drug developer, academia, physician, everybody would look at safety as the primary and the first question. So I would probably go there and say, hey, when it comes to determining safety, what my tolerable dose is, or the range is, see how effective simulation is. So that would be an initial, confidence enhancer. Then I would get to pharmacodynamics and then real efficacy.
How long would this process take before you're getting the FDA to start accepting the efficacy arm? If we followed your path,
Yes.
Is this a year from now? Is this 3 years, 5 years?
I would say more in the 3 to 5. Others might have different opinion, but I don't see this as a very quick thing because I would rather build it properly so that we build it and it stays and then really come and they don't leave. So that would be my
Okay, even with Rob Califf in the FDA head seat, it would still take 3 years. Okay, very good.
I was just going to ask, if you're going to try to build that kind of model for safety, I think you probably need different models for safety and efficacy that you have to build and train. But what would your input be? Would it be toxicogenomic data from cell line studies? I mean, what
I
do you
would
think
take mechanistic data, cell line studies. I would take preclinical data that we generate in order to enable an IND. I would take all of them and the mechanism I'm intervening, the pathway, and systems biology and everything to say how good am I in predicting a tolerable dose.
Yeah. I think, if I think about Sid's timeline, I think it's probably about right because you have to build it and then you have to demonstrate its efficacy even in predicting safety outcomes.
Okay, so 3 years, 3 to 5 years, very good.
After you build the model.
Gosh, are you going to help me out, Steffan?
Absolutely.
He's not helping me out. He's helping me out. So Lilly's on track to do 20 new drugs in 10 years at unprecedented speed. If you heard Ken Custer talk earlier, you heard him confirm that. And we are doing a lot of simulation to inform clinical trial enrollment. We do attempt to predict technical success of our trials. And we have a very large statistics organization that is instrumental in those efforts. We also have a very large real-world evidence organization, and we have had dialogue with the FDA. And I think where we're stuck is this chicken and the egg conversation. We have a lot of trials that succeed, and we have a lot of trials that fail, and we have a lot of in-between, right, to that rigorous subgroup classification point, whether it's ovarian cancer and a checkpoint inhibitor, lots and lots of other examples. And there's a set of pharma companies that have come together to put some of that data in a pre-competitive way in a solution called Vivli, V-I-V-L-I. And so I think what we have to do is change the conversation from one around what is the FDA going to do to how do we create that evidence that enables the FDA to have the framework, a better understanding of the AI, the methodological approaches, so that we can then get there faster.
Okay. Steffan, do you agree with that?
No, yeah, absolutely. I think from my perspective, in some ways, it's very clear what the FDA is seeking to accomplish. They're seeking to rigorously understand the benefit-risk profile of any drug so we can confidently bring it forward to patients with an understanding. Okay, what are the risks and what are the benefits? That's their goal. But number one is rigorous data.
Yep.
So the gold standard, the data that we need to bring forward, if possible, is randomized controlled data, randomized controlled trial data, and in oncology with overall survival endpoint. That's the starting point.
Yep.
Right? And then the extent to which you can perhaps deviate or not achieve that top tier, most rigorous data is all dependent on the unique situation. And the FDA, in my experience, has been very open to talk about unmet need. They don't want to withhold a drug from patients who could truly benefit simply because they're locked into some unrealistic data evidence generation. So that's number one. And number two, they understand that the expertise is out there in the world, and they're always willing to collaborate and work to understand how best to use it. So I think number one, pre-competitive collaboration, the FDA is always engaged, I think, in working with industry, working with academia, working with pre-competitive groups to understand how to move things forward. And when you come to real-world evidence, real-world data, synthetic control arms, in my view at least, that's a situation where we need to understand that the FDA is willing to actually utilize that data to make that decision, approval, non-approval decision, depending on the case. So very rare indications where they know there's no way you're going to be able to implement a randomized controlled trial. That's the setting where they're willing to look at, say, single-arm data. And if you can therefore bring forward synthetic control arm data to supplement that, they're very, in my experiences, they're very willing to listen to that as long as you've put a good rationale in terms of why it is you need to do that in the context of that specific patient population. So, I think that's the key to understand the right context. Because I can guarantee you, if you say you want to go into colorectal cancer and you want to have a single-arm trial with a synthetic control arm, it's not going to fly, right? So I think that's an important sort of perspective to have.
And you agree with the time frame of about 3 years before we're starting to see simulations being of the efficacy arm starting to take note?
Again, I guess it all depends on the application. I mean, maybe it could be even faster in the right unique situation. I mean, we had an opportunity to bring forward to the FDA an expansion of an indication where the data that we provided didn't have any clinical trial data. It was completely dependent on real-world data. And this was, what's the unique situation there? Where it was a drug called palbociclib that was already approved in ER-positive breast cancer, and the indication was in male breast cancer, incredibly small number of people, right? So the FDA agreed that, if you bring forward IQVIA medical claims data, if you bring forward Flatiron EHR data, incredibly small numbers by the time you target that very unique population, they were very willing to look at that data and actually expand the label. So again, it all depends on the application, and I think someone thinking in a forward-looking manner, maybe it'll be in 1 year, So I think it all depends on the specific context.
Yep. No, I could completely agree. Yep.
And I think some of that depends on, doing an intelligent filtering on the data, figuring out what the key variables are that you want to look at, being able to look at the available clinical data and pick out the right parameters so that if you did take it to a trial, you would be able to really identify those patients. And you could see sort of building on what Sid was talking about, doing the safety trials and trying to simulate that, and then taking the data that comes out of the safety trials to try to jumpstart the enrichment of the patient population going forward.
But I completely agree with Steffan's points that I don't think going in we should expect any resistance for the sake of resistance from FDA or any other health authority, but the onus is probably more on us to define the question, find the right data, and engage in a very productive conversation.
Well, this is definitely a different era that we can feel confident that the FDA will be a real partner here.
Before you, I just want to say, I think we have a good foundation for thinking about this because of adaptive clinical trials, right? How you update the models given the data. So this could really be an opportunity to sort of take that groundwork that's been done with the FDA and leverage it going forward.
That makes sense. Please.
Hi, I'm Michael Fitzhugh, managing editor at BioWorld. I just had a quick question addressing something Andrea talked about earlier, which was how models can inform trials as they go forward. Does that informing element sort of guide not only the potential successes, but also things that you try to avoid? I mean, are there trials that you model and you're like, whoa, that's not going to work at all. We're going to cut off that path.
I don't know the answer to that question explicitly, but I think it's highly likely that we do that, right? We have a very big effort around continuous learning. So when a clinical trial does fail, we hit that pause button, try and figure out, do we want to collect additional data, do additional sequencing, recreate that multimodal data set, and then we do team data sciences where we write down the methodology by which we're going to attack that problem and then do that retrospective analysis. And the intent is truly around, everything you've heard today, deep biological understanding, or is there something else that we needed to think about and do differently, all with the viewpoint to what can we take away from a learning perspective to the next trial. And how we operationalize that is, I think you're a program leader, but I have this huge book of computational work. And it's just a list of all of the projects and how are we going to prioritize and allocate resources to those projects from across all of the different disciplines at the company to make sure we do them with a very, very high standard towards rigor and quality.
Any other answers there as to when you guys decide, this is not a good problem or trial to bother model and simulate?
I think the point here is that as our understanding of the pathways and the network of pathways develop and our confidence in the simulation also improve, I can see a time when we would say mechanism X, run a simulation trial, don't bother going there. And that would be a benefit to all of us as well. Those no-goes are as important as goes.
Fail fast.
Yep.
Hey, Sandeep Patel. I work at BARDA, and we've been investing in in-Silico approaches and simulation approaches in a very different space in infectious diseases. And there's sort of two challenges that we're seeing, and I'm curious kind of how you're viewing this. One is that patient heterogeneity is a huge problem. How do you possibly simulate that or actually capture that in in-Silico? And the other challenge is just there are limitations to in-Silico models and thinking about, well, what else can you couple that with that would actually get you a good simulation? And we've been investing in things like in vitro tissue chips and things like that. So curious sort of how you deal with heterogeneity, especially with cancer, which is there's a huge issue there in terms of simulation.
Okay, anyone like to
Steffan, you want to
take
take
that?
that?
I'll make a quick comment. I think that represents one of the biggest challenges in how we can be successful in moving forward. It gets back to the issue of the data, because even a large number of patients, if you overlay the underlying heterogeneity, that becomes a small amount of patients. So the answer is, can you really scale up the number of patients? Because at some point, you'll encompass with enough N that heterogeneity that your models will be able to adapt and then utilize that heterogeneity. But I think it represents a huge problem in terms of just the data. We think we have big data, but it's not big enough for the kind of models I think we would like to generate.
Yeah, I mean, I'll just think, I think you need diversity of patients, right, in order to get to the diversity of data that you need. And that is a huge focus, especially in oncology. I always, share the story, breast cancer, we have a lot of women that survive, but the only friends I've lost to breast cancer happen to be from India, right? And if you look at where our genetic data comes from, it tends to be oversampled from European descent. So we've just started to partner with Refi Healthcare around increasing the diversity of patients in our clinical trials, and they are using AI behind the scenes because that'll eventually get to better representation that you need in order to get to the true solution.
Yeah, I mean, it's not just the right thing to have this greater diversity in trials. It actually is leading to better science and greater generalizability when you take these products into the real world. One shout out is to a group called the Global Alzheimer's Platform. And John Dwyer is in the crowd here. We announced 2 weeks ago a partnership between our company, GNS, and GAP. And this is the largest clinical trial of its kind in Alzheimer's with biomarkers and cognitive tests as the endpoint. But I believe it's 20% of the patient population that is African American and or Latina. And so it's really a big deal, and I think it's not just the right thing to do, it leads to better science. And then just one other aspect of that question, which is how do you deal with patient heterogeneity? It means you also need models that are down to that level of granularity in terms of genomics, right? I think the days of thinking that physiological models are going to be super useful in patient selection, especially in areas with great genetic heterogeneity like cancer, are over. But the good news is we have fast enough and cheap enough genomes such that the models, even though they're complex, they can incorporate those kind of mechanics.
But the genome is the starting point for a lot of this, right? And the point I made earlier with the genome playing itself out differently in every tissue. I think we have to collect that kind of data, but then we also have to be willing to simplify and to understand that the genetic factors that influence response in one tissue may be different from another. And we have to play this game, iterating between collecting massive data and then being willing to apply a biological filter to select what's relevant. And the other thing I think is really important that we've started to recognize is as scientists, we tend to be overly reductionist and think about all the genetic variants acting independently. But we really have to embrace the fact they're parts of complex networks that influence the outcome and work together.
Yeah, that's right. That's what kills us every time.
Yeah, hey, I'm Steve Wolf. I'm Executive Director of the Alliance for AI in Healthcare. So we're a advocacy organization of about 50 companies and universities, peers of yours. Interestingly, none of you all, yours companies exactly, but we'll work on that. And just to validate much of what I'm hearing has been our experience as well. As we were putting together our regulatory working group, we divided the work up into, AIML for, SAMD, the Digital Health Center of Excellence, which is easy because those algorithms are here and now and in product. And that's much easier for the FDA to wrap its minds around and to actually have something to regulate. But what we heard from, one of our leaders in particular who had been senior at the agency and then also at pharma and now heads up a nonprofit, he said, the FDA on the drug side is going to have real difficulty with AI. They'll be open, but they really do rely on the community to come to its own consensus and then for them to regulate around it. So, with that in mind, we started chatting with them, and kind of from their side, they were like, we've got the device and diagnostic side down, but we're really struggling on what they called horizon scanning. What's coming down the pike? And in terms of technology, in say the 3 to 5 year time frame, those were their words, so it just jived with what you had to say. Could you maybe think about doing a workshop where we could hear from some of the technologies that are new out there, coming down the pike, and then we would have sessions to talk about the regulatory implications of where they're headed. So we're in the process of putting that together. And so we were especially pleased to hear that, because this notion that, they're really going to rely on us to take the lead, we were concerned how they were going to respond to that. And here they were looking to industry and the community for that leadership. So, we'll be in touch to let you know how that comes together, because we're looking to go beyond our membership to get technologies to present and others like yourselves to be part of that, those panels to discuss. It's been a really useful engagement so far.
Great, thank you.
Hi, I think our conversation today have revolved around how to use the data. So my question relates to the data itself. So my question to the panel is, do you think we currently have all the technology we need to get data? Because it seems like the assumption is, oh, the data is already there, or you can get the data if you need it. But do you think that there are technologies that are still needed to collect the right data? And then my second related question is there any specific omics that you're excited about that you think can really help you in the near future to find the right patient, to find the right trials?
Okay, a great two-part question. Do we need new technologies that don't quite exist yet or aren't in use yet? And then where do you get the biggest bang for the buck in terms of any particular omic? So who would like to start the bidding?
So,
John.
There's an old saying when I was in high school that sometimes you have to dance with the one that brought you. And, we have data today, right? And there's no reason not to try to use it to its fullest advantage, right? We don't know what we might need that we don't already know of. So it's really important for us now to start with the data we have and see how far we can go and what kind of models we can build. As to your question about what the right type of data is, I think, again, it really depends on the situation. There's a really great article that appeared in MIT Technology Review about 1.5 years ago. And the title is, The Way We Train AI Systems is Fundamentally Flawed. And they talk about two problems. One is data shift, the fact that data collected here in San Diego might be different than in the Bay Area or in Boston or in Indianapolis, right? That there are biases in the data itself. But the other one, which is really important, is the problem of underspecification. And this comes back to really not being able to filter the data appropriately so you select the right type of features, right? Steffan early on, or Andrea was talking about imaging data as a surrogate for mutations. And we know those are related to each other. So we want to make sure we don't oversample by taking 2 or 3 or 4 different examples of the same thing, knowing that if we don't have one, we might be able to use the other. And that's where having expert input into this, into understanding this problem really becomes fundamentally important. So we want to try to leverage the data we have. We want to be on the lookout for other types of data. And I always say when we collect data, we want to collect as much as we can, realizing that for problem A, we're only using one subset, and for problem B, we might use a different subset.
I would just add to that is that with the fight over tissue already pretty intense, liquid biopsy, ctDNA, et cetera, where there's more development happening, I would love to see them progress further, imaging tools. And many tumors are not really amenable to biopsy, and serial biopsy, et cetera, they're not very practical. So that is a place, as a non-expert from the technology side, I would say there is room for development or need for development.
One comment that comes to my mind when you ask the question about data is I think oftentimes we don't have an adequate appreciation of the technical limitations of any given data that we collect. In other words, some people, they collect data, and if they're a computational biologist, that's all they want. They want the data, then they go to town on it. But I think we need to, even our computational biologists need to understand the limitations of the data so that the models they develop are taking that into account. I mean, one example is just transcript profiling. What we accept as adequate from the perspective of, say, whole transcriptome RNA-seq data is reproducibility, technical reproducibility with percent CVs of 20%. So then the question I have, okay, if you generate this model and you think you've got the best model, number one, validation, it's all about validation, right? Because most of the models are wrong, right? But so number one, validation, but also as part of developing the model, I know inherently some of the model approaches to developing models propose that they take this into account, but build in approaches, and I think this gets to the underspecification, that specifically address that issue. If your data is kind of semi-crappy, build into your model process a means by which you test the extent to which that data is going to be useful in generating a model. You know, for example, just rerun your model, introduce 10 or 15% random variation. Does your model hold up, or does it fall apart? So in other words, I think the methodology we use in generating models can really have a more informed view of sort of the end game, and also a view of the real world. What is this? And that's why I think that it differs so much from, say, a Netflix with an image, where probably the percent CV on that image is minuscule. So I think we have to realize this data is not the greatest data, right? And we need to make sure we take that into account.
Yep. So from the first question you asked, do we have what we need? You know, I always say people, process, technology. People right now, we don't have enough people with the right skill set, and we're all competing for them, right? And even practical example is I'd love more lawyers that understood AI and quantum, because it would make our ability to do some of the partnerships we need to do easier, right? So whether it's data engineers, data scientists, lawyers who understand AI, regulators at the FDA that understand a huge unmet need from a skill set and a people perspective.
Well, and data scientists to understand biology and the needs of the pharma
Exactly what
companies.
he just said, right? From the process perspective, we have so many guardrails, right? It'd be wonderful, I someday joke and say, to put the AI on the guardrails and tell me what guardrail applies depending upon the problem you're trying to solve, right? Because you can spend weeks, months trying to figure out what guardrails. And from a technology perspective, whether it's data or novel biology, right? The single cell, the long read sequencing, HTAs, microarrays, methylation. There's so many different types of sequencing. Sometimes I ask myself the question, what's the value of the $100 genome? Right? Can you give me an example of a project inside the walls of a pharma where the $100 genome is really going to help me? And I struggle sometimes with that. On the data side, I'd love more tools for generating the input for the AI/ML, like feature stores, self-services for the data platform. Making a movie and putting it on Netflix is a hell of a lot easier than, wrangling, imaging, TCGA, all of the different types of data that we are in the process of producing.
And the one thing we didn't talk about, which underlies all of this, but where we really need better definitions, is defining disease phenotypes, right? Where it's often a really squishy definition that makes building these models so much harder too.
Wow. Well, this has been a rather robust discussion. Let's just be clear on what we've learned. One is, I think this group, as a representative for the industry, is hopeful that these methods are making their way in industry and will soon be influencing the regulators. So that's a big deal. The estimates were 3 years, 5 years. Steffan was even more optimistic, said maybe even a year. So that's one. It's coming, I think, is what we're hearing. We also heard a lot of, I wouldn't call it skepticism, but a lot of focus on rigor in terms of the data and the AI methods and the aiming to the problem. I believe we heard that the biggest limitation at the moment is still high quality clinico-omic data, and we are limited by even the largest clinical trials in the world are only getting up to several hundred. Is that enough? Or do we need to supplement with real world data sets that are generating this kind of multi-omics? And so these are a few of I think the main themes we came out of this with. And I think my takeaway is that this industry is moving forward faster than not. I believe we probably had a good decade of where the data, the tools, the compute power was just not sufficient to do these things. The experiments are happening. The validations, the failures are going to be happening, and whether it's safety, efficacy, external control arms, and eventually external efficacy arms, the future, I think, is upon us. So please join me for a round in thanking our panelists.
Thank you. Thank you. Thank you.